Hey guys, in this video we will be discussing about HCL secretion. So there are three phases. First is cephalic. When we see the food, there is some secretion. And the second phase is gastric, when food is inside the stomach. Okay. And third phase is intestinal. And this is the phase when food is in the intestine. Okay. Now to proceed further, we have to know about the anatomy of stomach. So let's have a look. So this is our stomach. This dome shaped part, it is called fundus. And the larger and middle part, it is called body. And the last part that is near to duodenum, it is called antrum. Okay. So these are the major parts in the stomach. There is a mucosal lining inside the stomach, and the mucosal lining of stomach, it is folded inside. So it will look like this, because mucosal lining is folded inside, and these folds are called rugosities. These folds are called rugosity. Okay. In the mucosa, we have gastric glands. That secretes the HCL. So when we look in the detail, gastric glands, they will look like this. On the mucosal lining. Okay. So to study the HCL secretion, we have to study the structure of gastric gland. So this is how a gastric gland look like, and this is the luminal side, and this is the basolateral side. Okay. But by default, this is also lumen. So in the gastric gland, we have different kind of cells. The first cell, it will be called as ECL cell, electrochromaffin like cell. And this ECL cell, it will secrete histamine. The second cell, it is called chief cell. Okay. And the chief cell will secrete pepsinogen. And the last and most important cell, it is called parietal cell. And parietal cell will secrete HCL and intensive factor of Cahal. Now we have to look on parietal cell in detail. So this is the parietal cell. In this groove-like structure, they are called canaliculi. They are like canal, so they are called canaliculi. In the parietal cell, we have CO2 and H2O that will react and converts into H plus and a bicarbonate ion. And the enzyme that catalyzes this reaction is called carbonic anhydrase. The enzyme is called carbonic anhydrase. On the luminal side, we have a pump that is called hydrogen potassium ATPase. This pump will pump a H plus ion inside the lumen, and on exchange, it will get a potassium ion inside the parietal cell. So that's how we get our H plus ion inside the lumen. That's on the one side. But on the basolateral side, we have Also blood vessels, they will supply CO2 by the diffusion to the cell, because cell will be needing more amount of CO2 to generate more amount of H plus ion. And there is one more pump on the basolateral side, that is called bicarbonate chloride exchanger. Bicarbonate chloride exchanger, SCO3 minus chloride exchanger. So this pump exchange a bicarbonate ion towards blood vessel, and in exchange it will get a chloride ion inside the parietal cell, and this chloride ion, it will be diffused into the lumen from the luminal side by a channel protein, that is called chloride channel protein. Okay, so that's how we get our chloride and hydrogen ion, H plus ion inside the lumen, that is HCl. Okay, so this is the mechanism how we get HCl inside the lumen. Now let's have a look at its regulation part. So let's draw a parietal cell again. This is a parietal cell, and this is the luminal side, and this is the basolateral side. So there are G cells. There are G cells inside the stomach. They will secrete gastrin, and gastrin will act on CCKB receptor. CCKB receptor. It will stimulate CCKB receptor. And one more thing, as you know, the stomach is innervated by vagus nerve. Stomach is innervated by vagus nerve, and vagus will secrete acetylcholine, and this acetylcholine, it will act on M3 receptor. It will act on M3 receptor. And one more thing, remember we have talked about ECL cells and histamine. So this histamine, it will act on H2 receptor. It will act on H2 receptor that is present in the parietal cell. Okay, all three compound, they will stimulate hydrogen potassium ATPase. They will stimulate hydrogen potassium ATPase. So all these three, they will stimulate the H plus K plus ATPase. So you can say that the gastrin, ACH and histamine, they will positively regulate the HCl secretion. They will stimulate the HCl secretion. Okay, but we have some inhibitor also. So on the parietal cell itself, we have two more receptors, one for somatostatin, and second for prostaglandin. These two receptors, when they stimulated by somatostatin and prostaglandin, they will inhibit the H plus K plus ATPase. So you can say that the prostaglandin and somatostatin, they will decrease the HCl secretion. So gastrin, ACH and histamine, they will increase the HCl secretion. On the other hand, somatostatin and prostaglandin, they will decrease the HCl secretion. So that's all about the mechanism and regulation of HCl secretion. Thank you.